So hello guys, welcome to our channel Niperian Solutions. So today we are going to discuss about the previous year question paper of the Mpsc Drug Inspector which is held in the 2016. So here we are going to see a complete paper analysis and all the questions, whatever the technical questions that have asked. So let's start by one. So first one, HIV or AIDS is diagnosed by some specific test. So identify which among the following are used. Right, so what are the options? ELISA, DNA PCR by amplification, Western blot, and all of the above. So what is the answer here? That is the all of the above, because these all the techniques are used for the diagnosis of your HIV, right? But if they are asking which one of the following is a confirmatory test, so that is the Western blot. So Western blot is the confirmatory test for your HIV or the AIDS, and ELISA, DNA PCR by amplification, and even Western blot, these are the diagnostic test. But which one is the confirmatory? That is the Western blot. So that's why here correct answer is your option 4, that is the all of the above. The second question, what is the antidote for lead poisoning? So what are the options? That is the dimercaptosuccinic acid, physostigmine, fomepizole, and atropine. Right? So what they are asking? Lead poisoning antidote. So what is the correct answer here? That is the dimercaptosuccinic acid. What is the fomepizole is used in which poisoning? These are used in the alcohol poisoning or the methanol poisoning, right? So the next question, which of the following drug is not a tocolytic? Right? So what are the options? Ritodrine, magnesium sulfate, nifedipine, and ergotamine. Right? So question number 3, correct answer is your ergotamine, because your ritodrine, magnesium sulfate, nifedipine, these are examples of tocolytics, right? But what they are asking, please remember, they are asking not a tocolytic, right? Tocolytics are the uterine relaxant. So that's why correct answer is your ergotamine. Okay. So the next one. Okay, here are the classification of uterine stimulant and the uterine relaxant. So uterine relaxant are nothing but your tocolytics, and the question is from your tocolytics one. So please go through the classification of both oxytocic and tocolytics. So in the 2016 they have asked question on the tocolytics. So please go through it, and oxytocics also, right? So ergotamine, these are the oxytocic. So that's why our answer is ergotamine, which is not tocolytic. Okay, so the next one, which of the following NSAID is not a propionic acid derivative? So the options are ibuprofen, naproxen, oxaprozin, and piroxicam. So what is the correct answer here? That is your piroxicam, because these three are the propionic acid derivative, and here also they are asking the not a propionic acid derivative. So that's why piroxicam is the correct answer here. So the next question, which anticoagulant is produced by mast cell and basophils? So the options are heparin, prostaglandin, histidine, and vitamin K. So what is the correct answer? That is your heparin anticoagulant. So which is produced by the mast cell and basophils. So the next question, under which rule of Drug and Cosmetic Act 1940 and Rules 1945 license for blood bank are issued? So very important question, license for blood bank. So options are 122G, 122A, 160B, and 150B. So the correct answer is your 122G. Okay. So the next question. Okay, here are the classification of your non-steroidal anti-inflammatory agents. So here you can see that class propionic acid derivatives, right? So and piroxicam, pyroxine is your enolic acid derivative. The question coming from here only, right? Okay, next one, match the following. So here are different clotting factors, and they are asking the name of that clotting factor. So thromboplastin, proconvertin, fibrinogen, and prothrombin, right? So if you are looking to the numbering or the name of that clotting factor, here are the different options. So what is the correct answer here? That is the option number 1. Okay, means A, 2, right? So thromboplastin is factor 3, B1 proconvertin is factor what is 7, then fibrinogen, that is the factor 1, and the prothrombin, that is your factor 2. Okay, means option 1 is the correct answer of the question number 7. So the next one. Okay, here are the different coagulation factors with their name and number, right? So please go through it. What are the different names, like all the clotting factors? So please go through it, because this is very important topic, that is the blood. Definitely question is asking of the blood group or the clotting factors, which clotting factor is having highest molecular weight, which clotting factor is having the lowest molecular weight, right? What is the Hegman factor, what is the fibrin stabilizing factor? So please go through it, very very important topic, that is the clotting factors. Okay, the next one, basophils originate in. The options are megakaryoblast, monoblast, megakaryocyte, myeloblast. So what is the correct answer? That is your myeloblast. So basophils originate in the myeloblast. If you are looking like the synthesis of your all the like RBC, WBC, and all the platelets, so if you are looking to the chart, so definitely you will get the answer of this question. So this question is again from your human anatomy and physiology, or the pathology, that is the topic from your blood. Okay, then ninth question, match the following anti-cancer drug with their cell cycle effect. So this question is from pharmacology, anti-neoplastic agents or the anti-cancer agents, right? So they have given the different drugs with their cell cycle specific action. So which drug is acting on which phase? So that is very very important. So if you are looking to the drugs, so what is the correct answer here? That is your option number 1, right? Means A4, cytarabine, that is the RRC, that acts on a S phase. Then B1, etoposide, that acts on a G1 S phase. Then C2, vincristine, that acts on the M phase, and the bleomycin, that acts on the G2M phase. So these are very very important from the antineoplastic, which drug act on which phase. So here there are the different phase G1, S, G2 and M, and here are the drugs acting on the specific phase. So there are the two categories, cell cycle specific drug and the cell cycle non-specific drug. Means, so that's why the cell cycle specific drug, that are very important, and please go through it once. Next question, 10th one, which of the following is an ester and short acting local anesthetic? So the question comes from the local anesthetic topic, again from the pharmacology. So tetracaine, dibucaine, lidocaine and procaine. So here are the two different things. First one is the ester local anesthetic, and the second one is the short acting also, right? So firstly you have to remember which one is the ester and which one is the amide link. Okay, so the tetracaine, dibucaine, lidocaine and procaine. So among these one, your dibucaine and lidocaine, these are your amide local anesthetic, right? So that's why option number 2 and 3 are not the answer. So what is the remaining 1 and 4th, that is tetracaine and procaine. So what they are asking, the short acting local anesthetic. So which one is the short acting? That is your procaine, right? Tetracaine is not a short acting local anesthetic. That's why option number 4 is the correct answer of question number 10. The next one, antifungal antibiotic, amphotericin B is isolated from or the obtained from. Directly question asking from the microorganism and that source of that specific antibiotic, right? So amphotericin B, what is the source of amphotericin B? That is options are Streptomyces pimprina, Penicillium griseofulvin, Streptomyces nodosus, and both 1 and 3, right? So what is the correct answer? That is the Streptomyces nodosus, because if you are looking to the source of specifically all the antibiotics, so you will get the answer of this question. So Streptomyces nodosus, right? So amphotericin B is obtained from your Streptomyces nodosus. Question number 12, which of the following drug is not an enzyme inducer? So that is very important question from your general pharmacology, and what are the enzyme inducers, what are the enzyme inhibitors and examples. So that is also very important. So options are phenobarbital, phenytoin, rifampin and cimetidine. So what is the answer? As we know, the cimetidine is your enzyme inhibitor, not the enzyme inducer. Okay, and what they have asked, not an enzyme inducer. So cimetidine is not enzyme inducer, it is a enzyme inhibitor. So question number 13, one of the following drug is most widely used as a local anesthetic for infiltration, regional nerve block, epidural and subarachnoid anesthesia. Right, so they have given some specific features of the local anesthetic and what is the main word, most widely used. So as we know, what is the most widely used? So which one is that? Is the lidocaine, right? Lidocaine hydrochloride. So that's why option number 3 is the correct answer of your question number 13. Okay, here are the examples of ester link local anesthetic and amide link local anesthetic. So what is the trick here? If a drug containing single I, so these are your ester link, and if the drug containing the 2I, these are your amide linked local anesthetic. So similarly you can see in ester link, in cocaine only one I is there, procaine, chlorprocaine, tetracaine, benzocaine, only single I is there, but in amide linked local anesthetic 2I are there, right? In lidocaine, bupivacaine, dibucaine, prilocaine, ropivacaine. So every drug is having the 2I. So that is the very simple trick to remember ester link and amide link. And here is the second one, that is your short duration of action, and procaine is the answer, right? Because it is having a short duration of action. So that's why procaine is the answer of that question, right? Means like short acting as well as ester linked local anesthetic, that is your procaine. Okay, here are the microorganism and antibiotic. So what is our question? Like the amphotericin B they have asked. So amphotericin B, so what is the microorganism or what is the source? That is the Streptomyces nodosus. So similarly you have to remember that all the microorganism source for the specific antibiotic, Bacillus subtilis for the bacitracin, Bacillus polymixa for the polymyxin, like Streptomyces nodosus for amphotericin B, Streptomyces venezuelae chloramphenicol, Streptomyces aurifaciens tetracycline or chlorotetracycline, Streptomyces erythus erythromycin, Streptomyces griseus streptomycin, then Micromonospora purpurea, that is the gentamicin, Cephalosporium species cephalothin, Penicillium graciofulvin and the Penicillium notatum, that is your penicillin. Okay, so this table is very important for your exam point of view. This is from question from your microbiology we can say, or in even pharmacology also they have given the specific sources of the antibiotics. Okay, here are like general pharmacology, enzyme inducer and enzyme inhibitors. So cimetidine is the enzyme inhibitor, so that's why in that question cimetidine is the answer. Okay, so that is your enzyme inhibitor. Okay, the next one, which of the following antiretroviral drug belong to the non-nucleoside river transcriptase inhibitor? Simply they are asking the which one of the following is NNRTI. The options are nevirapine, indinavir, ritonavir, stavudine, right? So what is the correct answer? That is your nevirapine. That is the example of your NNRTI. Indinavir and ritonavir. So navir means these are the protease inhibitors. Okay. Then next question, question number 15. Which of the following drug induces delta amino levulinic acid synthetase? Okay, so the options are phenobarbital, ethanol, paraldehyde and diazepam. So what is the correct answer? That is your phenobarbital. If you are seeing the sedative and hypnotics topic from your pharmacology, so specifically they have given their barbitons or the barbituric acid derivatives. This induces the delta amino levulinic acid synthetase enzyme. Okay. The next question, 16th number. Which of the following drug is not a component of FDC used in RNTCP for TB? Okay, so the options are rifampicin, moxifloxacin, isoniazid and ethambutol. So what is the correct answer here? That is your moxifloxacin. Because if you are looking to the other options, rifampicin, isoniazid and ethambutol, these all the drugs are used in your tuberculosis, but moxifloxacin is not a combination, or that is the FDC combination used in your RNTCP. Next question, microorganism responsible for STD which causes an eye infection that is the primary cause of blindness worldwide. Very important question, right? So what is the options? Neisseria gonorrhea, Treponema pallidum, Chlamydia trachomatis, and Candida albicans. Right? So the correct answer here is your Chlamydia trachomatis. Okay? So that is very severe blindness due to your chlamydia, and it is also a sexually transmitted disease. Treponema pallidum, that is nothing but your source of your syphilis. Okay. Okay, here are the examples that antiretroviral drugs. So that's why classification is there, and here you can see the nevirapine, right? Nevirapine is our NNRTI, okay, non-nucleoside reverse transcriptase inhibitor. And what are the other? Navir are the protease inhibitors, right? These are the all drugs are the Navir. So Navir means protease inhibitor. So, and stavudine is from your NRTI, right here. So please remember NRTI, NNRTI, right? So this classification is also very important. So again and again I am telling you, so the classification from your pharmacology is very important. Please go through it, because definitely from each and every category or from each and every topic, at least 50% question are from your classification. And some of those are the mechanism of action, adverse effect. But if you go through the classification by heart, then definitely you are solving the questions of your pharmacology. So please go through the classification of whatever the topics they have given in your syllabus. Okay, the next 18th number, on which chromosome the gene for the 3 chains of the fibrinogen are found? Fibrinogen, right? So what are the chromosomes? Chromosome 1, chromosome 3, chromosome 4 and chromosome 6. So what is the correct answer? That is the chromosome 4, means option number 3. Okay. So the next, whole human blood is a mixture of blood and anticoagulant solution and contain not less than, right? So 8.7% hemoglobin, 2.3% hemoglobin, 9.07% hemoglobin and 9.7% hemoglobin, right? So please don't get confused between 9.07 and 9.7, right? Because all the options they have given in the weight by volume only. Okay, so the question number 19, the correct answer is your 9.7 weight by volume of hemoglobin. The next, match the following, right? So here are different drugs they have given. So like noradrenaline reuptake inhibitor, serotonin reuptake inhibitor, monoamine oxidase MAO inhibitor, then psychomotor stimulant. So if you know the serotonin, selective serotonin reuptake inhibitor, that is your fluoxetine, means B1. So where it is B1? That is in the option number 1. Okay, here option number 1 is the correct for this answer, because selective serotonin reuptake in is your fluoxetine. Okay, then psychomotor stimulant, you know amphetamine, it is the CNS stimulant. Okay, amphetamine, dextroamphetamine. So that's why option number 1 is the correct answer for the question number 20. So again this question is from your classification. Okay, because simply in that specific topic they have given the classification. Okay, so here are the classification of antidepressant. What are the selective serotonin reuptake inhibitors? What are the MAO inhibitors or typical antidepressant? So that's why I am saying please go through the classification of every topic which is mentioned in your syllabus. Okay, question number 21. The relative nephrotoxicity among the non-steroidal anti-inflammatory drugs is one of the following. So simply they are asking the what is the order of your nephrotoxicity. Okay, so indomethacin, phenopropine and ibuprofen. Ibuprofen greater than phenopropine greater than indomethacin, right? So that is totally opposite to option 1. Then option 3 is indomethacin is greater than phenopropine greater than ibuprofen. Okay, and then last one, phenopropine greater than ibuprofen greater than indomethacin. So what is the correct answer here? Question number 21, right? So question number 21 correct answer is your option number 3, right? Indomethacin is having the highest nephrotoxicity among your NSAIDs. The next one, question number 22. Which of the following antifungal drugs is used only topically? So the options are griseofulvin, clotrimazole, amphotericin B, and nystatin. So what is the correct answer? That is your nystatin. So nystatin is used only topically. So what is the accepted daily intake in mg per kg per day of aldrin? So options are 0.005, 0.008, 0.0001, and the last option is 0.0005. Okay, so the question number 23, the correct answer is your option number 3, that is 0.0001. So which of the following is non-opioid antitussive? Okay, so the codeine, ethyl morphine, dextromethorphan, and falcodeine. So the question number 24, correct answer is your option number 3, because codeine, falcodeine, and ethyl morphine, these are your non-opioid antitussives, but dextromethorphan is not. Okay, so that's why option number 3 is correct. Then match the columns. Again, this question is from your classification or the mechanism of actions. So phenothiazine, then atypical antipsychotic, butyrophenones, and lithium. And here are they have given the suppress inositol signaling, M receptor blockage, blockage of 5-HT2A receptor, and D2 blockers. Right? So for the question number 25, your correct answer is option number 3. Okay? Means A, phenothiazines, these are the D2 blockers. Okay? Then B3, that is the atypical antipsychotics, these are the blockade of 5-HT2A receptor. Then C2, that is the butyrophenones, are the M receptor blockade. And the D is the one, lithium is a suppress inositol signaling. Okay, so that's why option number 3 is the correct answer of question number 25th. Then 26, which one of the following drug is uterine relaxant? Right, means which of the following is a tocolytic? Okay, so in the last question also we have seen the examples of tocolytics, and the classification also we have seen. Right, so oxytocin definitely not, because it is oxytocic. Argonovin is also your oxytocic drug, means that is also not the answer. Okay, retodrine is the example of your tocolytic drug or the uterine relaxant. Okay, so that's why the option number 3 is the correct answer of your question number 26. Okay, the next one, the selective angiotensin 2 receptor blocker used in anti-hypertensive drug. Simple, very simple question. Sartans, right? So the captopril, verapamil, amiloride, and losartan. So what is the correct answer here? That is your losartan. The next one, what is the consequence of interaction between the 6-mercaptopurine and allopurinol? So the dose of 6-mercaptopurine is reduced, no effect. Dose of 6-mercaptopurine is increased, and the dose of allopurinol is increased. So what is the correct answer here? We know in the anti-neoplastic agent, because 6-mercaptopurine is having like that all the effects like hyperuricemia and all. So that's why dose of 6-mercaptopurine is reduced if given with your allopurinol. Okay, that's why option number 1 is the correct answer. 29, for donors experiencing plasmapheresis, aspirin should not have been taken days prior to donation. Okay, so options are 1 means like 1 day, 3 days, 5 days, or the 7 days. So correct answer is your 3 days. Next, which of the following oral contraceptive preparation contain an estrogen and a progestin in a fixed dose for all the days of treatment cycle? The mini pill, emergency pill, combined pill, and the phased pill. Right? So we know the combined pill contain the your estrogen and progestin in fixed dose. So the next one, 31, during and after an attack of infectious mononucleus, an agglutinin for sheep red blood cells appears in the serum and is of diagnostic significance. This agglutinin is demonstrated in the, right? So there are the different tests like Rose, that is the Rose-Oller test, differential sheep cell agglutination test, Paul Bunnell test, and Mg agglutination test. So 31 option number 3 is the correct for the question number 31, Paul Bunnell test. The 32 media that enhances the growth of particular type of bacteria and do not enhance the even may inhibit other type of organisms are called. The options are differential media, selective media, maintenance media and assay media. So maintenance media and assay media is not the answer, but definitely you are confusing between the differential media and selective media, right? But the correct answer is your selective media. Okay. 33. In the laboratory diagnosis of rabies, impression smear of the brain are stained by the Seller's technique to demonstrate the inclusion bodies called, right? So question from your microbiology. So that bodies are called as, the options are Negri bodies, Cowdry type A bodies, Cowdry type B bodies and Bollinger bodies. So the correct answer is your Negri bodies, right? Then Clawborough and Lilly's medium and Wills and Hope's medium are used for the cultivation of Bordetella, Clostridia, Bacilli and Corynebacteria. So what is the correct answer? That is your Clostridium species or the Clostridia. Okay. The next one, alcohol in water 60 to 85% is a good disinfectant for medical instrument and laboratory surfaces. It destroys the microorganism present thereby. The options are solubilizing lipid and protein denaturation, disrupting cell membrane, combining with SH group, acting as a strong alkylating agent. So they are just asking the mechanism how alcohol act to the disinfection. Okay. So what is the correct answer here? That is your solubilizing lipids and protein denaturation is the correct answer. Next 36, pH indicator or dye present in the Christensen urea medium, right? So neutral red, anhydride indicator, methyl red and phenol red. So what is the correct answer? That is your phenol red. Okay, the next one, which of the following statement are true? Okay, the Lal test reagent is obtained from a horseshoe crab. Okay, that is the correct statement. The Lal test is more sensitive than pyrogen test. Okay, that is also very correct because Lal test is very sensitive than your pyrogen. The next one, the Lal test only detect endotoxins of gram negative bacteria. That is also correct statement because Lal test determine the endotoxins of your gram negative only. And the last one, the Lal test can be used for radiopharmaceuticals. Means all the options are correct, right? So what is the correct answer here? That is your A, B, C, D. Option number 3 is the correct answer for this question number 37. Okay, the next one, the antimicrobial drug showing structural similarity to para-aminobenzoic acid, which is a bacteriostatic. Okay, so what are the options? Penicillin, rifamycin, isoniazid and sulfonamide, right? So if you are looking the structure, so that's why all the structures you have to know, right? So whatever the antibiotics they have given, you have to know all the structures. So penicillin, so this is the beta lactam antibiotic, so this is not relevant to your para-aminobenzoic acid, and it is a bactericidal, not a bacteriostatic. So that's why option A is incorrect, right? Because penicillin is bactericidal, because all the beta lactam antibiotics are the bactericidal. Then rifamycin, right? Then isoniazid. So these drugs are used in your anti-tubercular agents. Okay, isoniazid, rifamycin, right? So these are your anti-tubercular agents, but the structure is not similar to your para-aminobenzoic acid. Okay, so the sulfonamides. So sulfonamides are nothing but the your derivatives of your para-aminobenzoic acid derivatives. So that's why option number D is correct. Means which option is correct here? That is your option 4, D only. So sulfonamides are the only antibiotics which are really structurally similar to your para-aminobenzoic acid among these antibiotics. Okay, the next question, that is also very important question, very important. Please make a star mark, because bioassay of test organism and the vitamins. So last time also I said that one, the assay of antibiotics and assay of vitamins are very important. Means in the last time they have, in 2008 they have asked one question from there, and definitely in the 2016 also they have asked the question on the vitamin bioassay. Okay, so the lactobacillus casei, lactobacillus leishmani, lactobacillus aeruginosus and saccharomyces euverum, right? So we know the lactobacillus leishmani, so leishmani is vitamin like our B12. What is the vitamin B12, right? So this is our cyanocobalamin, means B1. Okay, so where is the B1? That is your option number 4 is the correct answer here, right? B1. So means A3, lactobacillus casei, that is your biotin. B1 that we have already seen, lactobacillus leishmani in the B12 vitamin. Then C4, lactobacillus aeruginosus, that is your calcium pantothenate. And the D2, that is your Saccharomyces euverum, that is your inositol. Okay, so please go through this topic guys, that is very very important. Okay, the next, the pyrogen test is designated to the limit the risk of the febrile reaction following parenteral administration of drugs. It is intended to be used for liquid products that can be tolerated by the taste dash in the dose of 10 ml per kg injected IV. So what are the options? Rabbit, white mice, guinea pig and hamster. So definitely for the pyrogen test what we are using, so that is your rabbit. What? That is your rabbit. So the next question, collodion filters are used for the isolation of bacteria, algae, molds and viruses. So question number 41, correct one is the, these are isolation of your viruses, collodion filters. The diagnosis of H. pylori can be done by urea breath test, copper breath test, sodium breath test and potassium breath test. So the correct answer is your urea breath test. The H. pylori, so these are again question from your microbiology, diagnosis of H. pylori, but definitely the drugs acting on the peptic ulcer, that is also very important. So H. pylori is there, which is responsible for your peptic ulcer diseases. So okay, here is the classification of the drugs for peptic ulcer, and in the last one here you can see the anti-H. pylori drugs. So the drugs, please remember these drugs: amoxicillin, clarithromycin, metronidazole, tinidazole, tetracycline and CBS. So please remember these drugs acting on the anti-H. pylori drugs. Amoxicillin, the only beta-lactam antibiotic which is active against the H. pylori, that is your amoxicillin, right? Clarithromycin, so this is your macrolide antibiotic. Then metronidazole and tinidazole, that is your that antiprotozoal and all drugs. The tetracycline, we know the tetracyclines, and CBS, that is the colloidal bismuth subcitrate. This act as an ulcer protective as well as anti-H. pylori drugs. So please remember the classification of the drugs for peptic ulcer and specifically the anti-H. pylori drugs. Next one, which of the following is used to serodiagnose syphilis? So again the diagnostic test. So in the first question they have asked, very first question in this paper, they have asked the serodiagnostic test for your HIV or AIDS, and here again the second question for the same category they have asked for your syphilis. So Venereal Disease Research Laboratory, Vidal test, Sereni test and E-test. So what is the correct answer here? That is your VDRL, that is Venereal Disease Research Laboratory, that is the serodiagnostic test for your syphilis, right? One another test is there for your syphilis, right? So that is your Kahn's test. Okay, so Kahn's test is also for your serodiagnosis of syphilis. The next question, question number 44, the biological oxygen demand values for the domestic waste water including the sewage are approximately dash units, right? So for domestic waste water including sewage, so what is the BOD? That is your 200. So option number 1 is correct here. The major cause of common cold, so that is again question from microbiology. So means pharmacology, microbiology, pharmaceutical jurisprudence, very very important subject. Okay, how many times I am telling you that please go through these subjects. So these are very important one. The major causes of common cold, right? Influenza virus, adenovirus, rhinovirus, none of the above. So we know the common cold, common cause is your rhinovirus. Okay, here are the viruses, okay, what kind of viruses and what are the infections associated with it. So please go through. In the last 2008 this question has been asked. What it is? That is paramyxovirus, mumps fever. Okay, paramyxovirus, mumps fever, asked in the 2008. Okay, so again here asking the common cold, that is the rhinovirus. So please go through these viruses and whatever the infection associated with it. Okay, the next one, which of the following statement about the antibiotic are incorrect? Please remember the word incorrect. Don't get confused between this one. Okay, first one, oral activity can be conferred on cephalosporin by esterification of the 3-carboxylic acid group. Directly question from your structural activity relationship. Okay, B option, acyl iridopenicillins are stable under acidic conditions. C option, aminoglycosides have a highly substituted 1,3-diaminocyclohexane central ring. And the D option, the enolized tricarbonyl methane system at C1 to C3 in the tetracycline structure need not be intact for good activity. Okay, so what they have asked, incorrect statement. So the first one, oral activity can be conferred of cephalosporin by esterification of carboxylic acid group. So means that is the correct one. Okay, but option B, acyl iridopenicillins are stable under acidic condition. So that is incorrect. And the aminoglycosides have highly substituted 1,3-diaminocyclohexane central ring. That is also incorrect. Means B and C are what is aminoglycosides have a highly substituted 1,3-diaminocyclohexane central ring, right? So means that is correct one. And the last one, the enolized tricarbonyl methane system at C1 to C3 in the tetracycline structure need not be intact for good activity, right? So the D1 is also incorrect because that is important for your activity. If you are looking the structure of tetracyclines, so please classify all the tetracyclines according to your structures like minocycline, oxytetracycline, chlorotetracycline. So definitely you will get the idea of the SAR. So this question is very important and very interesting from your structural activity relationship. Definitely these kind of questions we are not expecting in the exam, but please at least go once through your like medicinal chemistry or the simple points of your SAR, right? So for question number 46, the correct answer is your option number 3, that is B and D only. So B and D are the incorrect and A and C is the correct one regarding the antibiotics. So schedule M refers to, question number 47, what is schedule M? Pharmaceutical jurisprudence. So GMP and requirement of factory premises equipment for manufacturing of drugs, standard of surgical dressing, life period of drug and standard of cosmetic. So option number 1 is the correct because schedule M is regarding the GMP. Okay, then life period of drugs, that is schedule P, and the standard of cosmetic, that is schedule S. So please go through all the schedules from the pharmaceutical jurisprudence. Definitely one question is there. 1, 2, 3 questions are there from your schedules and the forms. Okay, the next one, who is the chairman of DTAB, right? It's a Drug Technical Advisory Board. So Drug Controller of India, the President PCI, Pharmacy Council of India President, and third one, the President MCI, and the Director General of Health Service. Very simple. The option number 4 is correct. That is DGHS, Director General of Health Service. Benzalkonium chloride, which is used as antimicrobial preservative, can be used in what concentration? So in last time also in the 2008 paper, the 2 questions are there from the concentration of your preservatives, and in the 2016 also they have asked the concentration of preservatives. So that's why if you are looking the previous year questions, you know the depth, you know the what kind of questions they are asking from the specific subjects. So 0.002 to 0.02%, 0.1%, 15% and 30%. So what is the answer? That is your option number A is correct. That is 0.002 to 0.02%. Question number 50. Practical application of recombinant DNA technology in the production of. First one, interferon, insulin, growth hormone and phospholipids. Right? So what is the interferon, insulin, growth hormone and phospholipids? So what is the correct answer here? That is your option number 4. Right? Option number 4 is correct, because phospholipids are not formed or not produced by your recombinant DNA technologies. Other interferon, insulin and growth hormone also produced by the our DNA technology. Okay, the next one. Which of the following statement of tautomerism is correct? Okay, so what about the tautomerism? Okay, so shifting of carbon atom from one hydrogen to another produce enantiomer is called tautomerism. Then that is the wrong, because this is not the tautomerism. Then next one, sugar with anomeric carbon undergoes the tautomerism in acidic condition. What is option number C? That is the sugar with anomeric carbon undergoes the tautomerism in alkaline medium. Means B and C options are very similar, just medium is changed, acidic and one is basic medium. Okay, so shifting of hydrogen atom from one carbon to another to produce enediol is called tautomerism. That is correct. Enediol, tautomerism or ketoenol tautomerism or enediol tautomerisms. So that is the very correct. So option number D is also correct and option number C is also correct, because in basic medium only that sugar with anomeric carbon undergoes the tautomerism in basic medium. So that's why C and D is correct answer of question number 51. So what is the options? Option number 4. Option number 4 is the correct answer. So 52. The most naturally occurring monosaccharides in mammalian tissue are of which configuration? Right? So that is very simple question. These are the D configuration, because L is not the natural configuration for your sugars. That is the natural configuration for your amino acids. All amino acids are L in nature, but sugars are the D in nature, dextro and levo forms. Okay, so dextro form commonly occur in the your sugars, and the levo form, that is your amino acids. Okay, the 53. Physical properties of well formulated suspension and emulsion are like the product must remain sufficiently homogeneous. Okay, that is very correct one, because suspension or emulsion, this should be a homogeneous. Okay, so the next one, the sediment or the creaming produced on the storage is easily suspended by a moderate agitation. That is also correct. Any suspended particle should be small and uniform in size. That is also very correct. The product needs to be thickened to reduce settling the red particles. Means they are talking about the viscosity, and that is also very correct. Means the question number 53, the correct answer is your A, B, C, D. That is your option number 4. Right? So all the options have been corrected. Okay. So the 54, the concentration of phenyl ethyl alcohol. Again one question is there. Means from preservatives, definitely one question is there. So what is the concentration range? So concentration range of phenyl ethyl alcohol they have asked. Options are 0.01%, 0.5%, 0.001% and 0.1%. Right? So question number 54, your correct answer will be 0.5%. Next one, which of the following is water soluble coating material used in dosage form? Which one? Solid dosage form. So water soluble. So options are gelatin, paraffin, shellac and nylon. So definitely your answer is your gelatin, because gelatin is the water soluble, because paraffin, shellac and nylon, these are the water insoluble. Ketone bodies in terms collectively called for. So they have given the different examples like acetoacetate, 3-hydroxybutyrate, acetone and ketone. So what is the correct answer here? Ketone is not a ketone body, right? Acetoacetate, 3-hydroxybutyrate and acetone, these are the example of ketone body, but ketone itself not as a ketone bodies. So that's why A, B, C is the correct answer here, right? So what is the correct answer? Option number A, B, C means option number 3. What are the factors affecting dosage form design? Right? So common ion effect, pKa, crystallization and autoclaving. Right? So factors affecting your dosage form. So I think all the options are correct, all of the above. Option number 4. Next, gamma aminobutyrate or the GABA is formed by the decarboxylation of, right? So the arginine, glutamate, tyrosine and spermidine. Very simple question. That is the answer of your L, glutamate. Okay. Next, hemolytic anemia can be caused by deficiency of following enzyme. Right? What are the options? Glucose 1 phosphokinase, 6-phosphogluconate reductase, glucose 6-phosphate dehydrogenase and transketolase. So the correct answer is glucose 6-phosphate dehydrogenase, means option number C only. So option number 3 is the correct here. Okay, because glucose 6-phosphate dehydrogenase is the enzyme which is responsible for hemolytic anemia, right? In antimalarial drugs, there is one drug which is causing the hemolytic anemia, that is your primaquine. Okay, the next, a drug shall be deemed to be spurious. Okay, so the question is from your pharmaceutical jurisprudence. So if it is manufactured under the name which belongs to another drug, if it has been substituted wholly or partly by the another drug or another substance, if purports to be a product of manufacturer or whom it is not truly a product, and if it contain any harmful or toxic substances which may render it injurious to health. Right? So which of the statement given above are correct one? Right? So if you are looking to the options, so this option A, B, C are the spurious one. But if it contain any harmful or toxic substances which may render it injurious to health, this is not a spurious drugs. So that's why option A, B, C is the correct one. Option number 4. Okay. Next, 61. Match the enteric coating polymers with their synonyms. So first one is the cellulose acetate phthalate, then shellac, then polymethyl acrylate, polyvinyl acetate phthalate. And here are the synonyms, right? So for the 61, correct answer is option number 2. Okay, because A1, because cellulose acetate phthalate is also known as surreteric. Okay, then B3, shellac is your aquacoat CPD. Then C2, that is the polymethyl acrylate, that is your certisil. And the D is the for polyvinyl acetate phthalate, that is your Colicot MAE. So that's why option number 2 is the correct answer for your question number 61. Okay, the next one, government analyst is appointed by state and central government under the section 33F, right? Because, okay, that is question again from your pharmaceutical jurisprudence. Very simple question. They have given directly statement like, so what are the options? Ayurvedic, Siddha, Unani and allopathic. So very simple, A, B, C. Because for like Ayurvedic, Siddha and Unani system of medicine, the government analyst is appointed by the 33F section, not for the allopathic. Means A, B, C only. So what is the correct answer? That is your option number 2. ASU system 33F is the government analyst. And what about the drug inspector? That is the 33G for the Ayurvedic, Siddha and Unani system. The next question, which of the following statement is correct with respect to the membrane controlled sustained release formulation? Okay, so release of the drug is by disintegration. A plasticizer is required to lower the glass transition temperature of the film. Fillers that compact by brittle fracture are suitable if used alone, and water soluble components are not necessary to increase the rate of release. Okay, so what is the correct answer? That is your option number B. A plasticizer is required to lower the glass transition temperature of the film. So because they are asking the correct one, right? What they are asking? Correct one. and that's why option B is correct. Means the option number 3, this one, only B is correct. Okay. Next, with respect to the suspension, the Stokes law particle is related to which factor? Okay. So if you know that Stokes law, what is the Stokes law? Sedimentation rate. It is telling about your sedimentation rate. So diameter of particle, density of particle and medium, viscosity of medium, and sedimentation rate. If you know that formula, like sedimentation rate is equal to, right, that sedimentation rate is equal to like the d square and density difference between 2 1, right, ds minus dm and upon 18n. So here is the viscosity, here is the density differences, here is the diameter and the sedimentation rate. Means that Stokes law is applicable to all the options given here, right? So that's why option number A, B, C, D. Which option is there? Okay, option number 4. Okay. The next 65 question. How many stereoisomers are possible for compound? So the compound has been given CH3, CH2, CH, Cl, CHBr, CH3. So simply you have to know, or you have to calculate how many asymmetric carbons are there. So if in this compound only 2 asymmetric carbons are there, and what is the formula for your isomerism? That is the 2 raised to the n. And what is the n here? N is nothing but your asymmetric carbons, and here is the n is equal to 2, means 2 to the power 2 is equal to 4. So that's why 4 isomers are possible in given compound. So option number 3 is the correct answer for the question number 65. So which of the following is not an anionic surfactant? Means given they have below the examples of anionic surfactant except one. So the options are carboxylic acid, alkyl and alkyl aryl sulfonate, ammonium carboxylate, and substituted alkyl amide. So carboxylic acid and sulfonate, that we know these are the anionic. So ammonium carboxylate or the substituted alkyl amide. So among these, which one is your cationic? Cationic surfactant is your option number 3, that is your ammonium carboxylate. Right? So that is the correct answer, which is not a anionic surfactant. So the next, the control test for gauging of surgical catgut is carried out at. So they have given the relative humidity 45 to 55% and temperature 30 degree. Then RH 45% and temperature 25 degree. Ambient temperature and humidity. Then RH 60 to 80 degree. RH 60 to 80% and temperature 16 to 21 degree Celsius. So for 67, correct answer is your option number D, that is the relative humidity 60 to 80% and the temperature from 16 to 21 degree Celsius. So the question number next 68. According to the schedule U, the particulars shown of manufacturing records for substances other than parenteral are. Okay. So question number 68. The options are serial number, name of the product, lot or batch size, reference of master formula record. Right. So and what they have asked means like correct they have asked. So serial number definitely it is include. The name of the product, batch size, lot size, a reference of the MFR, so that all the things are included. Means option number A, B, C, D. So that's why your option number 4 is the correct one regarding the question number 68. Drug shall be deemed as misbranded. Okay. If it is not labeled in the prescribed manner. Okay, so that is correct one, because whatever the drug not labeled in the prescribed manner means the misbranded drugs. If it is colored, coated, powdered or polished so that damage is concealed or made to appear of greater therapeutic value than it really is, so that is also misbranded drug. Okay. Next, then if its label or container bears any statement, design or device which makes false claims for the drugs, so that is also your misbranded drugs. If it is prepared, stored or packed under the insanitary condition, so that is not your misbranded drug. So that's why A, B, C is the correct for the misbranded drug. So that's why option number 2 is the correct answer. Okay, the next one. In continuous fermentation, steady state is achieved using principle of thermostat, Levy-stat, pH stat and chemostat. Right? So correct answer is your chemostat. The important disaccharide are sucrose, maltose, lactose and cellulose. So definitely cellulose is not an important disaccharide. So that's why your A, B, C are the important disaccharides. So which option is this? So that is your option number 3. Okay, sucrose, maltose and lactose. Okay, very important, this misbranded drugs, adulterated drugs and spurious drugs, their definitions, what are the points covered in the misbranded drugs, adulterated and spurious. Please go through this slide, please take a screenshot of this slide, because definitely in the coming exam also at least one or two questions are there, because in this 2016 paper they have asked 3 to 4 questions on the only this slide. One question is from misbranded drug, one question is from adulterated drug and one question is from spurious drugs. So please, we are expecting in this coming paper also, so please go through this slide, please take a screenshot, very important. Okay, here are the important disaccharides, sucrose, maltose and lactose. So please remember the hydrolyzed product also. Sucrose is like glucose and fructose, maltose is the 2 units of the glucose and the lactose, galactose and glucose. And please remember the linkage also. And sucrose is a non-reducing sugar, right? Sucrose is non-reducing sugar, that is also very important. And the non-reducing sugars does not undergo the oxazone formation reaction. Please remember, right? These does not undergo the oxazone formation reactions. Okay, next, according to D&C Act 1940, Schedule M2 is meant for the manufacture of cosmetics, medical devices, pharmaceutical products and allied products. Right? So what is the correct answer here? That is your cosmetics. Because Schedule M is related to your good manufacturing practices. And again it is divided into M1, M2 and M3. So M1 is related to the manufacturing of your homeopathic products. M2 is related with your cosmetics, manufacturing of your cosmetics. And M3 is related to your manufacturing of your medical devices. Please remember M1, M2 and M3. Accordingly, in Schedule F is also again divided F1, F2, F3, FF. So please go through this F schedule among all the schedules. So the next one, which of the following statement relating to DNA vaccine is correct? So which one is the correct one? So the pathogen gene itself is used as a vaccine, either naked or inserted. Okay. Gene coding on immunological protein, then polysaccharide vaccine and the attenuated non-pathogenic. So what is the correct answer? That is your option number 1. Next, identify the incorrect statement for an adulterated drug. Again one question is there from the adulterated drugs. So you know how much importance you have to give to your pharmaceutical jurisprudence, specifically spurious, misbranded and the adulterated drugs. So what are the options? It purports to be a product of a manufacturer of whom it truly not a product. Okay. Then it is not labeled in the prescribed manner, that is your misbranded drug. It is substituted for another drug, that is your spurious drugs, and it bears a color other than prescribed. And what they are asking? They are asking the incorrect statement. Okay. So for the question number 72, option number A, B, C. Okay. That is the correct answer for the question number 74, because A, B, C is incorrect for your adulterated drugs. Okay. Okay, the next one. In sustained release dosage form, the drug release is retarded due to the presence of Okay, then special coat, embedded matrix, core tablet and granule mix. So here is the correct answer is your embedded matrix. The next one, the time period to supply information under the Right to Information Act, that is also mentioned in your syllabus. Right to Information Act, life or liberty of person. Okay, so what is the correct answer here? That is your 48 hours. Okay, so the next one, list of essential drug include, right? So nucleoside means NRTI, T-zidovudine and its combinations, insect repellents, that is diethyl toluamide and DDT, then anti-Parkinson medicines, levodopa and carbidopa, and plasma substitutes, that is the tranexamic acid. So what are the options, right? A, B, C and D. Okay, so among these, list of essential drug include. Okay, so these all the drugs are included in your list of essential medicines or the list of essential drugs. So that's why for question number 77, option number 1 is correct. The next one, the following pairs given GMP requirement as per the various schedules. Which of the following pair is incorrect? So schedule T, then GMP requirement of Ayurvedic, Unani and Siddha medicines. Okay, that is correct one, because schedule T is about the ASU GMP requirement. M1, just we have discussed, M1 is regarding your homeopathy preparations. M2 is about cosmetic preparations, and schedule N is of your running pharmacy. But here requirement for manufacturing of medical devices. No, that schedule N is not the requirement for manufacturing of medical devices. That is your minimum equipment running for pharmacy, that is your schedule N. And what is the option they have given? Requirement for manufacturing of medical devices, that is incorrect. That is for your schedule M3. Okay, so that's why option number 4 is incorrect here. Next, the present Drug Controller General of India. So this is the question paper from 2016. So that question is not important right now, but you have to know who is the Drug Controller General of India in this means right now who is the DCGI of India. So who is this? That is the Dr. Rajiv Singh Raghuvanshi. Who is DCGI of India right now? That is the Dr. Rajiv Singh Raghuvanshi. At that time Dr. G.N. Singh is there, but right now please remember Dr. Rajiv Singh Raghuvanshi. Question number 80. The key principle for the regulation of prices in the National Pharmaceutical Pricing Policy 2012 are follows. Okay, the options are essentiality of drugs, control of formulation price only, market based pricing, cost based pricing. Okay, so cost based pricing is not there, so option A, B, C are the correct. So that's why option number 2 is the correct answer here. Consumer Protection Act 1986 is applicable to all of the following area, right? So Maharashtra only, Madhya Pradesh only, Gujarat only, and all over India except Jammu and Kashmir. So the option number 4, right? It is applicable to all over India except Jammu and Kashmir. When was the minocycline drug belonging to the category of antibiotics cleared and approved by the DCGI New Delhi? Right, so that is also very old question, but please remember the options are June 1975, Jan 2015, Jan 1988, and then May 1988. So what is the correct answer? That is your May 1988. The function of the CDL, that is the Central Drug Laboratory, in respect of the classes of the drugs are enlisted below. Okay, so laboratory in respect of blood grouping and diagnostic kits for hepatitis B, C virus, the National Institute of Biological, Noida. Laboratory with respect to VDRL antigen, that is the Pasteur Institute, Kannur. Then Indian Veterinary Research Institute, Hafkin, Parel. And the laboratory for homeopathic medicines, that is the Ghaziabad. So they are asking which of the following pair are true. Okay, so for the 83, option number 3 is correct, that is A and D only. Okay, means laboratory in respect of blood grouping and diagnostic kits for hepatitis B, C virus that is National Institute of Biologics, Noida, and D, that is the Laboratory for Homeopathic Medicines, that is the Ghaziabad. Okay, the 84th question. Advertisement of drug used in following diseases and disorder are prohibited. So the drug for procurement of miscarriage, drug to correct menstrual disorder in women, drug to improve fairness, drug to improve the power of human being for sexual pleasure. Right? So there are the different options. So what is the correct option, or what is the prohibited, right? So drug to improve fairness, that is not the correct one. Otherwise A, B, C, right, these are the correct statement. Okay, so that's why A, B and D. So that's why option number 2 is the correct answer for question number 84. The National Cancer Registry Program, which is a part of a National Cancer Control Program, was initiated in the year 1982 by which following organization? Okay, so very simple question. AICT, UGC, ICMR and Tata Cancer Research Center. So correct answer is your Indian Council of Medical Research. The main regulatory authorities which use guidelines that are globally accepted followed by the pharma industries are, okay, so US FDA, UKMCA, ICH and WHO. So these all are the very important. So that's why all of the above. Question number 86, answer is all of the above. The function of CDL in respect of any drug of the class are carried out at, okay, so Central Research Institute Kasoli, prescribed laboratory by central government, ISO certified laboratory and NAC accredited laboratory. So option A and B correct. So that's why option number 2 is the correct. Central Research Institute Kasoli and the prescribed laboratory by the central government. Okay, the next one. Functional foods are termed as a nutraceuticals. Which one of the following is an example of nutraceuticals? So isolated compounds, dietary supplement, design foods and processed foods. So for question number 88, all are the examples of your nutraceuticals. Means option number 4 is the correct one. Narcotic drug include, okay, the narcotic drug include coca leaf, acetylorphin, cannabis and ketobimidone. So these all are the narcotic drug. So option number all of the above, that is your option number 3 is the correct answer for question number 89. For import license, which of the following statement is incorrect? Okay, so import license in form 10A to import drug is specified in the schedule X. So they are asking the incorrect. Please remember, they are asking the incorrect statement. Import license in form 10 is to import drug excluding those specified in schedule X. So that is also correct, and option number 1 is also correct. Then an application for import license shall be made in form 8 for drug excluding those specified in schedule X. Okay, so that is also correct. And the option number 4, an application for import license shall be made in the form 9A for drug specified in the schedule X. So that is the wrong answer. Okay, because that is the form number 8, not 9A. The 91, the permitted synthetic organic colors and the natural colors in the cosmetic shall not contain more than 2 ppm of arsenic, 20 ppm of lead, 2 ppm of mercury and 100 ppm of your heavy metals. Right? So for question number 91, correct answer is your A, B and D only. Right? A, B and D only. Poison Act 1919 mention, right? So what it is? The grant of license to possess any specified poison for sale, wholesale, retail and fixing of the fees to be charged for such licenses. The classes of person to whom alone such licenses may be granted or sold. The maximum quantity of any such poison which may be sold to any one person. The maintenance of recorded by such vendors and the labeling, packaging and safe custody by person who said or possessed it for sale. Means that all the options are very correct. So that's why A, B, C, D or all of the above is the answer. So option number 3 is the correct answer for question number 92. 93, the drug will be deemed to be spurious. Again one question on the spurious. Means I think 4 questions from the adulterated, spurious and misbranded drugs. 4 to 5 questions are there. So it is imported under name which belongs to another drugs, right? Then it is colored, coated, powdered or polished so that damage is concealed. If it is not labeled in the prescribed manner, if it is made to appear of better or greater therapeutic value. So means they are asking about the correct statement about the spurious drug, right? So only A is the correct regarding the spurious drugs. So that's why option number 4 is the correct, that is the only A. So these are the all the questions asked in the 2016 paper from the technical one, and only 7 questions are there from the non-technical intelligence test, right? So means we are expecting in the coming exam also only 7 or max to max 10 questions are from the intelligence test. So if you are looking the whole paper, definitely you will get the idea how much what is the depth of the question paper or from which areas they are asking the questions. If you are looking the paper, then definitely you will come to know that the questions from pharmacology, specifically classification and mechanism of actions, then pharmaceutical microbiology, and then pharmaceutical jurisprudence. So most of the questions are from these 3 subjects. So please prepare for the drug inspector exam accordingly, and please go through your syllabus. Thank you so much guys. Thank you so much.